I am reading my scholarly paper titled Safety, Tolerability, and Efficacy of Creatine Supplementation in Huntington's Disease. Creatine is an amino acid that is produced naturally within the human body. Creatine that is found naturally in the body is formed in the liver, pancreas, and kidneys. Once creatine is created in these organs, it is then transferred to the skeletal muscles of the body. This creatine is then transformed into phosphocreatine. Phosphocreatine plays an essential role in providing the phosphate necessary to form ATP, and ATP is the molecule that is considered the energy source of a body. Having large amounts of phosphocreatine in the body can help quicken the recovery of ATP during high-intensity exercise. Creatine supplementation provides an influx of phosphocreatine in the body, therefore improving the performance of the high-power activity that is dependent on this energy complex. The supplementation builds up a supply of phosphocreatine in the muscles that can be utilized quickly. Due to this system, creatine is often used as a supplement to improve performance of high-intensity exercises. The supplement may also benefit individuals with heart disease or muscular diseases. Because of its recognized activity and muscular activity, creatine supplementation is being considered as a supplement that can delay the symptoms that occur with Huntington's disease. Huntington's disease is considered a neurodegenerative disease that results from a mutation of the Huntington protein. This then leads to a variety of symptoms, both physical and neurological. The disease causes involuntary physical movements, cognitive decay, and psychological issues. Currently, there is no treatment to prevent the disease, but therapy that utilizes various dietary supplements is under investigation. It is thought that such therapy may ameliorate the symptoms associated with the disease. Creatine is one dietary supplement that is being studied as a method to help reduce or delay the progressive symptoms of Huntington's disease. As discussed earlier, creatine helps to provide cellular energy. Huntington's disease results in mitochondrial damage, and the damage affects the cell's energy system. If such energy is necessary for proper functioning of cells and systems, then it is thought that a supplement which can boost energy levels may help improve neuronal functioning in Huntington's disease. Ferrante et al. Investigated the possible neuroprotective effects of creatine in the mouse animal model. The findings reported in this study suggest that creatine acts in stabilizing intracellular calcium and inhibiting activation of the MPT, both of which are crucial to prevent cell death. When creatine is administered to the body, it provides numerous benefits to the functioning of the body's energy system, as well as reduces injury to the DNA. When evaluating the benefits of dietary supplementation, it is important to consider the potential risk associated with such therapy. The relative risk for creatine use can be evaluated by determining the number of adverse events that occur over the period of supplement use. A review of the literature in regard to safety and the use of creatine in individuals with Huntington's disease suggests that when given in appropriate dosages, individuals taking the supplement do not experience significant adverse effects. A study conducted by Hirsch et al. 2006 supports this generalization. The study considered both the safety and tolerability of creatine in Huntington's disease. In the study, subjects were either given a placebo or 8 grams of creatine monohydrate per day for 16 weeks. The results of the study indicated that the 8 grams of creatine daily were tolerable. The incidence of adverse events was similar between the placebo and the test group, and the differences in the occurrence of any particular adverse event were insignificant. This research supports for the use of creatine for individuals with Huntington's disease. Another study conducted by Tabrezi et al. Investigated the outcome of the administration of even higher dosages of creatine. This study focused on individuals with Huntington's disease as well. Each participant within the experimental group received 10 grams of creatine per day over a period of 12 months. This high dose of creatine resulted in very limited adverse effects. Only mild nausea and diarrhea were reported as adverse effects among the experimental group. Consequently, these studies indicate that supplemental creatine, even in relatively high doses, is safely tolerated by individuals with Huntington's disease. While there is appropriate evidence to suggest that supplemental creatine is safe when ingested in doses of 8 to 10 grams a day, there is still a need for further research that focuses on the administration of even higher dosages of creatine. The tolerability of creatine supplements is another therapeutic parameter that must be examined. Supplement intake must be tolerable for patients to be able to complete the designated dosage levels over time. The use of creatine in Huntington's disease appears to be quite tolerable based on several studies. One study conducted by Hirsch et al. Examined the tolerability of creatine supplementation. The study evaluated tolerability by determining the number of treatment failures, patients who did not complete the study, in the group given creatine in comparison to the placebo group. The results of the study indicated that the treatment given, which was 8 grams per day of creatine, was well tolerated. The investigators reported that out of approximately 15 subjects enrolled in the study, 5 individuals could not complete the study. 3 of those patients were from the creatine group and 2 were from the placebo group. The study also revealed the incidence of adverse effects and found no significant difference between the groups evaluated. Therefore, the treatment given did not affect the likelihood of study completion, and the creatine therapy was deemed well tolerated by patients. The study by Bree et al. Also reported that tolerability again did not seem to be an issue. Out of the 13 patients with Huntington's disease who participated in this study, only 2 patients were unable to complete the full term of the study. Thus, this study suggests that the use of creatine supplements in individuals with Huntington's disease is well tolerated. Although the literature suggests that creatine therapy is safe and tolerable, the efficacy of this dietary supplement in individuals with Huntington's disease is limited. There currently is not enough clinical research on humans to strongly evaluate the efficacy of this supplement. Studies done with animal models, such as the Ferrante et al. Study, do indicate that there is evidence of efficacy in animal models. The outcome of that research indicates that creatine administered dose-dependently did increase the mouse survival rate. It also revealed that neuronal shrinkage and gross atrophy of the brain were delayed in the affected mice. However, while at least one study completed with Huntington's patients has indicated that creatine supplementation decreased the amount of circulating biomarkers consistent with deterioration in neuronal health, definitive human studies regarding the efficacy of therapy are lacking. Research studies completed by Bree et al. And Ryu et al. Support the safety and tolerability of creatine, but the studies were unable to provide considerable evidence of the slowing of symptoms. Bree et al. Was able to indicate that creatine was increased in areas of the brain and muscle. Other research trials demonstrate similar results, but again are not sufficiently powered to dictate whether creatine can delay Huntington's disease. Due to this lack of research in humans, I would assign the efficacy symbol of NR to creatine supplementation in Huntington's disease. With the research I have reviewed, I can be a source of knowledge to consumers regarding creatine supplementation. Specifically, I now understand the role of creatine supplementation in Huntington's disease. After reviewing research studies and clinical trials, it is clear that the efficacy of creatine in Huntington's disease is still unknown. As discussed, there is confirmation of its effectiveness in animal models, but research is lacking in humans. This points towards the need for further clinical research in humans to establish the true performance of creatine within this disease. These studies also warrant further research in determining the appropriate creatine dosage, as the superlative dosage of creatine for affected individuals has yet to be established. While creatine's efficacy is still unknown, the evidence from numerous studies indicates that the supplement is both safe and tolerable for individuals with Huntington's disease.